Let's start SAP Lumira. So here we have already created a ChakraView SAP Lumira extension. We'll load this LUMS file. Mm, yeah, of course, it'll take some time. Yes. So finally we have our data set ready over here. Now this data set contains a list of chromosomes, and then it contains the start and the end position for the gap as well as the size of the gap. So this data and another data set called mRNA information, which is also imported, and it contains the data set from the real world that we have just mentioned. It came from the ShimerDB. So this data set contains a lot of information about the genomic fusion that has happened between different cancer cells. Let's say there is this mRNA sequence, and there is this chromosome 18, and into this position, 218 up till 218 the chromosomes, the bases located from this position have been translocated to this position, which is present in the chromosome X. So this kind of a translocation is trivial in the sense that they signify certain amount of information, and this information is basically present in the annotation database provided by UCSC, NCBI, or several other cancer data management institutes. So once we get to know about the data distribution, the relationships among different mutations, we can drill down to just select the few of our interested mutations and just to validate against the annotation databases. So once the data is in here, let's start the visualization. So currently we have a visualization where we have our ChakraView selected over here. Currently we are just dealing with chromosome number 11 and chromosome number 13. These are forming the start or the head ends of the chromosome. So we have two different colors, the green and the dark green, and they have all these things, these Bezier curves, each one of them stating a fusion from starting with the chromosome 11 into each of the different chromosomes. So let's see if I'm interested to see a mutation that is starting from chromosome 11 to chromosome, let's say 5. Then I'll just pick this curve and click on it. So it will give me this information about the entire gene fusion. So I could, from this graph, I could actually see that the length of the chromosome is actually distributed across the arcs of the circle, and each of this starting position is in proportion to the actual start position of the gene fusion. So if I see any arc emerging out of this, then I could just say that okay, this fusion is just located at the end of the sequence, and maybe of higher interest compared to those that are present in the center. So this kind of data is very useful for the researchers and biologists. Normally this kind of data is hard to visualize with current visualization techniques, and whereas a circular vision technique provides a handy tool for such kind of visualizations. And moreover, we have this interactive fashion of point and click, where we can have the information about the entire sequence, or the entire fusion, and from this information, actually we can look at the annotation databases, or the biologists can look at the annotation databases and get the relevant information about the kind of mutation that has taken place between these two chromosomes. Let's say I just select a different chromosome over here. Let's say I just want to see about 17. So yeah, so I have all the gene fusions about 17. looks from the graph that most of the fusions happen across like let's say for chromosome 1 there are a lot of fusions happening from 17 to 1. Yeah, so I have created a few more graphs of interest, and this is for the chromosome M, X, and Y that are also of prominent interest to the researchers. So yeah, and finally I would like to say a graph like this, which contains almost a lot of chromosome information, and in this since it is very cluttered, it makes sense to just say see the link over here. Currently this actually a different kind of a graph, which just says the connection between the start chromosome to the end chromosome. So such kind of different visualizations can be, or let's say different variants of the circular visualization can be done. So the data set is essentially the same, but the implementations are essentially different. So we have measures and dimensions, and all these dimensions I have preloaded just to save time. Another visualization, let's say we just go to the storyboard, and yeah, there are some storyboards that I've created previously. Yep, a multi-view. Yes. So so the biologists can actually have a very handy tool with them in order to work with this kind of data. I mean, this is not actually restricted to the bioinformatics domain, but you can use this tool to actually analyze any kind of relation. Let's say each of these individual arcs can represent a table, or can represent columns in a table, and then columns among different tables, and then you can just plot a relationship between those two columns. So basically this is very generic. You can actually take up any data set and just plot it. So another gap analysis was the use case that I mentioned previously, and we'll be working with this data set for that. And I'll just go to the visualization. Now this gap analysis, as I told previously, helps in validation, because normally there are certain tools for alignment, and this kind of tools should actually not consider the positions where gaps are actually present in the microarray sequence. So let's say if the gap, if I'm interested in chromosome 15 and there is a huge gap over here, I'll just hold over it, and it says that from starting from 0 to this position there is a huge gap, and normally I need to actually not take the results that are thrown from sequence processing, which falls into this range. So let's say I take up this gap, and yeah, it's hard to pin, but yes, this for the chromosome 11. So this is a start and the end position for that particular gap. So normally the graph could be actually made more intuitive, but let's say for the sake of data visualization in circular fashion, we have created this new technique currently, and we are in the process of putting more and more or improving, let's say improving this ChakraView by putting more and more features into it. So we have one storyboard for this graph called gene gap analysis. Yeah, so so this kind of visualization technique could really prove to be a very handy tool for the researchers and biologists. Yeah, so by providing such a technique inside Lumira, one can actually just share the graph created, and they can actually publish their results also into the research papers, because this kind of graphs provide a lot of information in very small space. So it becomes very handy tool to just use it for the publications also.